because the evolution stuff you do on your own in there. And the evolution stuff, the cumulative final at the end, the evolution is just that one text, and then you never have to do anything with it again. Oh no, chapter 12 is included. Thank you for reminding me. Well, it's going to be on your test, so it. So what we covered in the last chapter was mitosis, your non-sexual reproduction, what goes on. So what we covered in the last chapter was mitosis, your non-sexual reproduction, what goes on with your somatic cells. It's everything that is not the sperm and egg. This section looks at meiosis in the sexual life cycle. So this is how the sperm and egg are going to reproduce, which is a little bit different in there. And this starts to tie into genetics in their inheritable features, because you're looking at how do things get passed down from one generation to the next. The somatic cells in there, they just make clones of themselves, but your sex cells combine with somebody else's sex cells to make a new organism that's a combination of the two. So heredity is just the transmission of traits from one generation to the next, and genetics is the study of hereditary and the variation that are in there. So offspring are going to acquire their genes from their parents, and they do that by inheriting chromosomes in there. The gene is going to be the unit of heredity in there. It's going to program for specific traits. The gamete's going to be the sex cell or the reproductive cell. So in animals, it's your sperm and egg. It's also, you have sperm and egg in plants. It's just, you don't, it's a little different with plants in there. I just have a hard time thinking about plants and sex. It just, even though I've known about it for a long time, you stand still, that's not sex to me. But that's beyond the point. We eat their ovaries, yes, of your plants. That's what fruit is ovaries. Lettuce? I knew there was fun going on out in the yard that I was missing out on. So your somatic cells, these are going to be everything else except the gametes, and they're all going to have 46 chromosomes in them in humans. The locus is the specific location of a gene along the chromosome. So when we're talking about its location, we call it its locus or loci. So in asexual reproduction, you get an offspring that's the exact genetic copy, which is a clone in there. Clones are just groups of genetically identical individuals, where with sexual reproduction, the two parents give rise to an offspring that's got its own unique genetic setup in there that's inherited from the two parents. So when we look at the life cycle refers to the generation to generation sequence of stages in there. So basically from conception to making its own offspring in there. When we look at the chromosomes, one way you can look at them is through a karyotype, which is an image of the chromosomes that's going to be arranged. And what they do is they arrange them in pairs, so they're with their homologous chromosome in there. And they put them in order from biggest to largest in there. So this is showing for humans in there. When they do this, they usually enlarge them and they match them up. On Canvas, there's some links to a website that has, like, where you can go in and it gives you all of these, and you can drag the chromosomes to match them up with each other online, so you don't have to cut them out on little pieces of paper and match them up if you're bored. You're not going to have to do that in here, but it just kind of gives you the experience of, oh, this is what they do with the karyotype. So if you were to look at a karyotype and there is an extra chromosome in there, you know there is a problem. If there is a chromosome missing, you know there's a problem in there. And the karyotype isn't going to give you a whole lot more information other than extra chromosome or missing chromosome in there. So we have the pairs of homologous chromosomes that are duplicated here. So you've got your sister chromatids here, and here you've got the centromere in there. So homologous chromosomes are two chromosomes that compose a pair. They're going to have the same length, the centromere position, and they'll have the same staining pattern. So you can see they are matched chromosomes in there. So a sex chromosome is going to be one that's going to determine the individual's sex in there. XX is female, XY is male. And only a small part of that chromosome is actually homologous in there. The Y chromosome is a lot smaller than the X chromosome. So when you look here, you've got this big X and then the tiny little Y chromosome on there. All the rest of them are autosomes, so those are non-sex chromosomes in there. When we talk about how many chromosomes a cell has in there, how many pairs, they're either going to be diploid or haploid, at least when we're looking at animal cells. In plants, you can have polyploidy, where you can have multiple copies of the same chromosome, and it doesn't seem to be a problem for them, but that doesn't work in an animal cell in there. So the diploid cell is any cell that's got two chromosome sets in there, and the way we abbreviate it is being 2N in there. So humans, their diploid number is 46 in there, so you have 23 pairs of chromosomes. Haploid cells are going to be your gametes. This is when they have half of the genetic information in there, so they only have a single set of chromosomes, because that cell is going to meet up with a single cell of chromosomes from somebody else to make the new zygote that you'll end up with. And we represent that as just a single N for haploid. So the fertilization is when you have the union of the gametes that's going to result in fusing of a nuclei. So you have two cells that fuse together to become one in that process. And then after that happens, those are able to go on and do just plain mitosis to continue to develop. So the zygote is that fertilized egg in there. So when you undergo meiosis, you're going to reduce the number of chromosomes getting ready in this process. So there is different types of sexual life cycles in different organisms in there. You have the alternation between meiosis and fertilization that's common to all organisms in there, but what we look at is the timing of the events and at what part of their life are they going to be multicellular, what part of their life are they just single-celled in there, and that's how we come up with this little diagram at the bottom that is explained up here. So in your animal cells, the multicellular organism is going to be diploid. The only time you have haploid cells is your gametes in there. Otherwise, all of the rest of your cells are diploid in there. And so the gametes don't go any further through any further division prior to fertilization. So in this picture, it's showing here that you've got your gametes being your haploid stage here. And then in the multicellular stage here, from the zygote on up to making your gametes through meiosis, it's diploid in that phase of the cycle for animals. In plants, it's a little bit different. They go through this alternation of generations where they can be multicellular in both the diploid and haploid states in there. So the sporophyte is going to be what we call the multicellular diploid stage. So after they do meiosis, they can produce these haploid spores, which will go through mitosis then to make a multicellular organism that is haploid. So it would be like if sperm started to reproduce on their own and made a multicellular giant sperm-like creature. So it just, it makes the whole plant way of doing things just weird. I'm definitely animal person, not a plant person here. So that's what you'll get with the sporophyte state. Then with the gametophyte state, you have the multicellular in the haploid stages in there. So it gives rise to the gametes. So for plants, what you have is multicellular haploid and multicellular diploid in there. So they do a mix of both. And then we get to the fungi and some of the protists, and these will be haploid unicellular multicellular organisms. Their gametes fuse and form a diploid zygote in there, and then meiosis occurs without making diploid offspring in there. So this diploid stage is only a small part of their life cycle, and the majority of their life cycle is going to be in a haploid state in there. So it just depends, are you animal, plant, or fungi, how you do the dominant part of your life cycle in there. So with plants where you have this sporophyte and gametophyte generations in there, you can actually get like two different versions of the way a plant will look in some cases in there. So horsetails are a really primitive plant that's been around for a long time, that they grow all over the place. If you have horsetails in your yard, you're not getting rid of them anytime soon. And they will, sometimes they come up and they're just a little pointy thing on the end. I'm not going to draw it on the board because it just looks obscene the way I draw it. And other times they come up and they look bushy like trees. And it's the same plant, it just has two different phases. What I would say is this summarizes it. The picture is a lot easier to summarize that than I think using the words are to summarize the difference in those. So with meiosis, you're going to reduce the number of chromosome sets from being diploid to haploid, and there's two stages of meiosis that you're going to go through. With meiosis, you do two divisions and you end up with 4 daughter cells at the end in there. So we've got the sister chromatid cohesion. This is going to be where you have your 2 copies of one chromosome and one duplicated chromosome that are going to connect together in there. The homologous chromosomes are the individual chromosomes that we inherited from the different parents. Those can have different alleles on them, and the allele is the version of the gene. So what this is showing here is an interphase. Basically, you've got these homologous chromosomes here that will pair and make up the sister chromatids, and that you've got 2 copies of them there. So in meiosis 1, you separate these 2 copies into 2 different cells in there. In meiosis 2, you're going to split those copies into 4 different cells here. So each of those ends up with half the genetic information. So does that make sense how you start out with this cell here? You copy your genetic information to get ready to go through cell division. First division, you put 1 copy in each, and then each of those divide, and you put 1 copy in each of those cells in there. So if you compare what goes on with mitosis and meiosis, in here you've got the duplicated chromosomes, and it just goes through 1 division and you end up with 2 copies of the chromosomes in each of the cells. Here it's going to go through a 2nd division, which is just going to put 1 copy in there. So there's some events that are unique to meiosis in here, where you have the synapsis and crossing over. So you've got these duplicate homolog pairs in there that are going to form this synaptonemal complex where you're going to have crossing over occur. And so if you've got your 2 legs of the chromosomes here, what's going to happen to increase your genetic variation with the crossing over in there is you're going to have genes swap which of these they're located on. So if you didn't have that, what you would get is this one goes one way, this one goes the other way. But what's actually going to happen to increase the variation in there is you're going to have part of it switch places on there to increase the variation. So if this is, say, it's gene ABC here, it's going to be gene ABC here that will switch places in there. So it isn't that you're going to end up with, 3 different genes for eye color over here and none over here. If you have one gene for eye color, it trades places with another gene for eye color on the other side in there. So they will also line up as homologous pairs on the metaphase plate instead of as individuals. So they're going to line up, it's just you've got pairs instead of individuals in there. And then you'll have the separation of the homologs in there, where the duplicate chromosomes or the homologous pairs separate out, but the sister chromatids stay together in there. So what's going to allow them to stay together is they've got this cohesion enzyme in there that holds them together, and they're not going to be able to come apart until anaphase 2 in there that will release it to completely let it come apart in there. So there's just a different enzyme that's involved in the 2 different states. So as you're going to form the spindle in there, you're going to have the chiasmata that's going to hold the homologs together for the first mitotic division, and then the cohesion gets released to separate in the second one. So we have 2 different types of divisions that will go on in there. We have one of them that is the reductional division in meiosis 1, where you have the number of chromosome sets in there, and then equatorial division in there in meiosis 2, where the sister chromatids separate in there to produce the haploid cells. So what makes the sexual reproduction much more interesting is the genetic variation you get out of it in there. So with the clones, it's not all that exciting. You just get the same thing over and over and over, and it's exactly the same. You might get a mutation from something in the environment, but otherwise it's pretty predictable. You're going to get the same exact thing. However, if you go to make a baby, you don't really know what you're going to get out of the deal. You're taking a chance in there. So the independent assortment of chromosomes. So when you go to divide the parental chromosomes, you have one chromosome you got from your mom, one chromosome you got from your dad. When you go to make your sex cells, you don't know which one is going to go to which cell, but those have to divide up. So it's going to be independent assortment. You may send mom's chromosome 1, 2, and 3, and then dad's chromosome number 4. But each chromosome has a 50-50 chance of ending up in each of the 2 different cells in there. So it ends up being what we call independent assortment of the chromosomes in there. And that will occur with every different chromosome in there. So for humans, you have 2 to the 23rd chromosomes in there, which means without crossing over, just the variation of different sperm or egg you could make by dividing the chromosomes alone is 8.4 million different ones. So if you think that, you know what, I really liked this first kid and I'd like to get another one that's like it, you're going to have to try a whole lot of times to get the same traits again. It's just not likely going to happen in there. So the odds of you creating 2 kids that look exactly alike in there, while yes, in theory it's possible, you can't make that many babies in there. So as a female, you're limited on how many you can carry. I don't think you could get that busy as a man to make that many babies in there either. So this is showing here where you can get these different combinations in there. This is showing, you know, possibility 1, possibility 2 in here. And so you've got 4 different combinations just from looking at those few chromosomes in there. So with crossing over, it's going to produce a new recombinant chromosome that's going to be completely different in there. And so it's derived from two different parents, your 2 different parents when you make it. But it's still going to be a unique chromosome in there. So each chromosome has on average 1 to 3 crossover events. So this is showing it with just 1 crossover event, but you may have 3 in there. You could have more, you could have less on there. Average is 1 to 3 in there. And those will cross over at specific proteins precisely corresponding in the exact same location on the other chromosome in there, so that you end up with the recombinant chromosomes. So the chiasmata, this is that location where you're going to have the crossing over occur in there. You've got the centromere here. And this shows how you get those different combinations in here. So this is showing it with 2 different points on this picture. So in anaphase 1, this is what you end up with. After anaphase 2, this is what you end up with. And now you can see you've got these daughter recombinant chromosomes in there that are all different in there. So when we look at fertilization in humans, each male and female gamete is about a 1 in 8.4 million possible combination in there. And then when you look at the fusion of 2 zygotes in there from 2 different partners, you have that 2 to the 23rd times 2 to the 23rd ends up with 70 trillion combinations that you can make just between 2 people. So if you don't like the kid you got from somebody the first time, you'll get something different the next time out of them. So the evolutionary significance of the genetic variation in there within the population, with natural selection, you want to accumulate more genetic variations, because if you have more variations, you're more likely to have one that's going to survive better in the environment in there. And that's what natural selection is going to favor, is the one that survives in there. So the mutation will produce different alleles. So that's one way of getting more variation in there. Also with the sexual reproduction is going to create more genetic diversity in there. So during times when the environment is stable, it actually will favor asexual reproduction in there. It's more advantageous in there because they know there's something that's successful and working. That's more advantageous to asexual reproduction. When the environment is less stable in there, sexual reproduction is more favored in there because it's going to give you more possibilities that will be able to adapt to that unstable environment in there. In animals, you have almost exclusively sexual reproduction. They don't necessarily adapt, they just die. To survive with me, a plant needs more ability to adapt than what nature has given it. But when some of the plants, the reason you can take starts off of them is it has to do with sporophyte versus gametophyte. It doesn't, if you're not in a sexual reproduction phase, you can just take a part off and it will basically be a clone of itself in there. It doesn't have to have the pollen and the fertilization in that phase.
Well, same with asexual reproduction. Like if you are able to give it off of yourself and your offspring, how can your offspring adapt?
Well, they don't adapt as well. It would have to have mutations occur in there. Yeah, because a lot of times when you just are copying the DNA, you'll make a mistake. and that will introduce variations in there. So one of the reasons why animals have been fairly successful is the sexual reproduction has allowed more variation in there. When you, like with dogs, if you look at dogs, they tend to be, mutts tend to be healthier with less problems than where if they've taken purebreds and they've only bred them within a limited population, because there's less variation introduced in there. In the purebreds, if there's a gene for a disease, it just keeps getting passed on and passed on, where when the dog just goes and mates with whatever it can find out there, which is about what they do, it's a lot more genetic variation that gets introduced in there to the species. So horizontal gene transfer, this is when you can have DNA from another species come in to without it being passed on to the next generation. So one example are rotifers. They'll dry up, their membrane cracks, and then the DNA from other rotifers and other species comes in through the cracks and gets incorporated in. Bacteria are going to have more horizontal gene transfer in there for genetic variation. You're not going to have a lot of that occurring. You don't really have any of that occurring naturally with humans in there. It would have to be artificially induced in there. And that's the end of chapter 13.